All right, section 6.4 is talking about and focusing on the endomembrane system, what its component parts are, and its job or jobs. So when you talk about the endomembrane system, we're talking about a few organelles and parts of organelles specifically. We're really talking about the traffic system, the system of transport in the cell. So that includes the nuclear envelope, the ER, the Golgi, the lysosomes, the vacuoles, and then the plasma or cell membrane. They are not all continuous. Some of them are, like the nuclear envelope and the ER are continuous, but then between the ER and the Golgi, the way that those stay connected would be through vesicles, which are very small vacuoles that transport material. When you talk about the endomembrane system, the ER is really one of the most important parts of it because it is really the, well, like your book calls it, the factory that produces materials that that the cell sometimes has to keep, but then also a lot of it is bound for export out of the cell. So the ER is very large, makes up, like it says, more than half of the total membrane in eukaryotic cells, and it is continuous with the nuclear envelope, which means it's part of it. It branches off of the nuclear envelope. And there's two parts to the ER. You got the smooth and the rough. Smooth doesn't have ribosomes on it, but the rough does. And so there is a drawing as well as an electron microscope image of it, so you can see the difference between the smooth ER and the rough ER. You can see the ribosomes studying the surface here of the rough ER, and you can see that clearly over here in this area there aren't any ribosomes at all in the smooth ER. And you can see where it's continuous with the nuclear envelope, right in here is where it's connected, as well as a few other locations. Okay, so what does the smooth ER do? Well, the smooth ER has some jobs. It synthesizes lipids, right? Smooth, think smooth ER. Lipids are kind of smooth, right, and oily. So it makes lipids that are going to be used either in membranes or transported out. It does help with the metabolism of carbohydrates, helps breaks those down. It also helps detox poisons, so metabolic poisons that are produced by the cell, as well as poisons that are brought into the body that have to be broken down and detoxified. That's the job of the smooth ER. And then you'll also find that it stores calcium, and calcium is important in the firing of neurons and the neuromuscular junctions and the contraction of muscles. So calcium is a very important ion in cellular function. Now the rough ER, it's rough once again because there's ribosomes attached to the outside, and its job is that it helps secrete glycoproteins, which are proteins bonded to carbohydrates. These things usually end up on the membrane serving as usually ends up on the membrane acting as a communication device for the cell, or a signal, or a way for cells to recognize whether it's the cell or organelles with those glycoproteins on it. The products that it makes are passed from the rough ER to the Golgi by transport vesicles, which are basically just membrane packages that hold the proteins to get them from point A to point B. And then, the rough ER makes a lot of the membrane. That's its main job. Now, the Golgi apparatus will take, it's like the shipping and packaging center, right? So its job is to take the products being made by the ER that get sent from the ER to the Golgi via those vesicles. It takes those, it receives those, and it does several things to it. But the Golgi is really just a bunch of membranous sacs that we call cisternae. And so those membranous sacs, the materials fuse from one side, the vesicle, fuses with the ER side of the Golgi into the cisternae, out of it, and into the next, and into the next, and it gets processed as it does that. So what happens to it while it's in the Golgi? Well, it will modify the products. It will make some macromolecules as well, and then it packages materials into transport vesicles that are marked with the right signals, the right protein signals on the membrane, so that that vesicle knows where to go. So it knows, okay, this vesicle has to get to the plasma membrane so that this product can get, you know, exited out of the cell via exocytosis, or maybe this, you know, transport vesicle knows, okay, it's marked to go someplace else in the cell. Right, so here we have an image of the Golgi, right? You see the cisternae, right, which are the, you know, areas within, right, within the sacs there. But then you also have two sides of the Golgi. You have the cis, or the receiving face, and that's where you can see the vesicles coming in. And then those vesicles will just move from one to the next to the next to the next until they bud off of the trans face. Think the trans face is like transport, right? This is the end of the Golgi that's going to transport the vesicle out to wherever it's going in the cell, okay? And then we have lysosomes, the next component of the endomembrane system. Lysosomes are specialized vacuoles composed of or containing hydrolytic enzymes that fuse with food vacuoles or other vacuoles holding maybe waste. And they have hydrolytic enzymes that will just break down, right? Hydrolytic, using water to break down, right? That's what that name means. So they go in and they will help break down food vacuoles and other waste products that aren't needed. And they can hydrolyze all macromolecules. Now, other things that happen then with the lysosome, it says some types of cells can engulf another cell, right, using phagocytosis. Phagocytosis is just the engulfing of large pieces of matter, and that will then form that food vacuole. So like think about like how an amoeba gets food, right? We know what an amoeba looks like, right? That little blobby, weird shaping guy, right, with a nucleus. But as it sees a food vacuole or a piece of food in its environment, it will extend these pseudopodia out, right, until eventually it surrounds it, and now this guy is in a food vacuole inside the body of the amoeba, and that will fuse with a lysosome and digest those molecules. It can also recycle things in a process called autophagy, which basically means self-eating, and so it will destroy components of the cell itself to recycle them when they get worn out. And so here we see a lysosome fusing with a food vacuole here, right, combining with it and, you know, using the process of cellular digestion, and then those waste products will then be, you know, passed along to the cell membrane and spat out, gotten rid of. And then we also have, you know, when we have worn out organelles like the mitochondria, right, it will be surrounded by a vesicle along with a peroxisome, which helps detoxify the cell and get rid of waste that are produced during mitochondria or during cell respiration by the mitochondria. But the lysosome, when it's worn out, it will fuse once again with that vesicle holding those things and break down those products so that, you know, we can get rid of worn out cell parts. And vacuoles, they have a lot of different jobs. There's lots of different types of vacuoles. You can have, you know, in a plant cell, you have food vacuoles, sure, and waste vacuoles, and vacuoles that contain worn out organelles, but you also have that central vacuole, which is really important in plant cells, that maintains that turgor pressure inside a plant cell, right? That pressure, that water pressure that makes a plant cell, you know, be full and look healthy. So you can have lots of different types of vacuoles doing different things. So food vacuoles formed through phagocytosis or the engulfing of food. You have contractile vacuoles that you can find in freshwater protists. Now, because remember about water flow across the membrane and hypotonic and hypertonic and isotonic environments and what they do to the cells that are in them. And so a freshwater protist in fresh water is like a cell being in a hypotonic environment. So the water is going to want to come into the cell, and if it didn't have any way to get rid of the excess water, it would explode because all it has is the cell membrane to protect it. So contractile vacuoles pump that excess water out. And then we already mentioned the central vacuoles that you're going to find in plant cells that hold you know, water and organic compounds, salts and things for the plant. Right, so there's an image of the central vacuole, like a big water balloon in a plant cell. Big and taut will keep that plant cell upright and healthy, and when that central vacuole emaciates or withers or loses water, dehydrates, then the plant cell starts to, be a little bit shriveled up, and that's what contributes then to the overall health of the plant. And so when we talk about the endomembrane system, remember it is the system within a cell that produces and transports materials from start to finish. And why is the nuclear membrane included? Well, because that's where the messenger RNA exits out of the nucleus. That messenger RNA goes to the rough ER, to the ribosomes found there, gets produced into a protein, and then that protein gets sent to the Golgi where it's modified and packaged into a vesicle and then sent out of the cell. Right? So in a review here in terms of how does it work, there's the nucleus with the ER attached to it, right? The products get spit out of the